today to open the 2015 edition of the Sam Idison Lecture. And this lecture is now in its 23rd year and was started in honor of Sam Idison, who in fact was the inaugurating chair of the Interdepartmental Graduate Program in Neuroscience. And a couple of years ago, 23 years ago, it was decided to honor the memory of Sam Idison by installing this lectureship, which every year is given to one of our very best graduate students. Now, all of our graduate students would deserve to be up here and give a lecture, but unfortunately we can only have one of them present this lecture, and the honor this year goes to Dilrub Parikach. And it will be wonderful to hear his lecture. I just wanted to say a few things about Sam Idison before we start to sort of keep his memory alive a little bit. So he was born in 1918 in Buffalo, and then came in 1938 to UCLA to study in the chemistry department. So he was one of the very first graduate students, undergraduates, I think, at UCLA. It took him a while, so he started in 1938. He graduated in 1947 with a BS in chemistry. And it didn't take him that long just because he was a lazy student. He actually was fighting in the war in between with the US Army. So like many people of that generation, they actually lost a lot of time in their education then. But he came back to UCLA afterwards, got a PhD in chemistry in 1952, working on biochemical metabolism, the metabolism of bioactive compounds. And very soon afterwards, after he finished his PhD and started his faculty position here at UCLA, he wrote the then most important textbook on biochemistry and behavior. So he started looking, how do these biochemical pathways actually metabolize things in our brain? He started in about 1961. He was one of the co-founder of the Brain Research Institute, started to work on monoamine oxidase, or MAO, a molecule that he actually worked on until his retirement in 1987. And then he published his last paper in 1990, so just 15 years ago. He was one of the very influential people, I think, that we have here at UCLA. Neil Parikach, I always get that wrong. I practiced and I still get it wrong. Double majored at Rice University in math and in biochemistry and cell biology. And both of these subjects, as we will hear today, have actually accompanied him ever since then. He joined after an early stint in the neuroengineering program, where he was still an undergraduate at Rice. He came to UCLA, worked in imaging labs for a while, a very productive time to him, but then decided to join the MSTP program, and after 2 years of medical training, joined Dan Geschwin's lab. Neil, as you will see, is an absolutely outstanding researcher. The letters of recommendation were over the top. And just to sort of give you an idea, he got an NRSA, he was an E32 training grant, he has 2 Cell papers, a Nature paper, a Nature Neuroscience paper. So as a now 5th-year graduate student who just defended his thesis, but hasn't handed it in yet. As a graduate student, he published 9 papers. In the letter of recommendation, it said 7. So I thought, wow, only 7. So I just looked him up, and actually, since the letter of recommendation was written, there are 2 more papers that appeared. One of them was in molecular human genetics, and the other one in Nature Neuroscience. So he's not kind of quieting down, so just having had an absolutely fantastic career. Being an MSTP student, he's going back to medical school in a month or so and finishing up his medical training. So we're going to lose him for a little bit from science. I think he will be a loss for science and for neuroscience, but hopefully we can entice him to come back and continue his fantastic work that he's going to tell us today about. So congratulations for being the Idison Lecturer, and it comes actually with a cash prize. that I don't have here, but that I will give you to, that hopefully was given to you, and the little plaque that you can hang in your office or at home.
Can you guys hear me? Great. Thank you, Felix, for that wonderful introduction. And thank you, everyone, for coming to my talk here, despite the fact that some of you might have come to my thesis defense just 2 months ago. I think this is the first time I've had a picture of me that's much larger right next to me. That's weird. But anyhow, what I'll be talking to you today is about insights into the molecular underpinnings of autism from the analysis of gene expression in human brain. And this work was done in Dan Geschwind's lab, and it'll cover several different topics. But first of all, what's autism spectrum disorder? So autism is an intersection of two behavioral phenotypes. You have deficits in social communication and restrictive and repetitive behaviors. And these symptoms must arise developmentally. The deficits must result in significant social, occupational, or other life impairments. And they should not be better explained by some other disorder, like severe intellectual disability. However, this autism spectrum is pretty common, and 1 in 68 individuals in the U.S. are currently have autism, and about half of these individuals also have intellectual disability, so an IQ less than 85. So it's intellectual disability, but not so severe that it explains the deficits. And most of these individuals are diagnosed based on symptoms that occur before the age of 3. So what we have here is a neurodevelopmental disorder that has a lot of comorbidities, but these two share components. And the and the comorbidities include epilepsy, ADHD, language impairments, and other comorbidities, including hypersensitivity, several other things that I won't go into. Now, the last 2 decades have been a revolution in autism because it's always been debated what causes autism. Well, we know now that genetic risk factors contribute heavily to autism. We know that because of twin studies. So monozygotic twins are 36 to 95%, so this is just all the studies out there, the range, concordant for a diagnosis of autism, whereas dizygotic twins are much less concordant. And if you look across the studies, the average really ends up being about 70% for monozygotic and maybe 20 to 30% for dizygotic, suggesting a strong genetic component. Additionally, if you have one child with autism, it increases the chance of having the second child with autism from that 1% background rate to about 15%. So this all points towards genetics. So there's been a large effort over the last decade to figure out which genes are involved in autism. Now, to understand that, we have to understand genetic variation in the human population. So all of us have a genome. We have 3 billion bases in our genome, and there's different types of variation in the genome. So if you have 1 point mutation, that's called a single nucleotide variant. And there's about 3.7 million of these in your genome. This is where the idea comes that 99.9% of your genome is the same as another person's, 0.1 differs. But you also have insertion deletion variants, so there's about 360,000 of those, and they are either more than one nucleotide that it either pops in or pops out relative to the general population. You have copy number variants, which are longer swaths of the genome that can range anywhere from 1,000 bases to a megabase or even more, and the average individual has about 700 plus of those, and exceptional individuals have even more. And by exceptional, I mean not in a good way. For autism, the main difficulty right now is to figure out what are the risk variants versus chance variation, because you have all this variation that occurs in people anyways, right? And there's many different study designs that have been used, and there's many different genetic models that have been proposed. And the basic story that's coming out of these, people have used genome-wide association studies, they've used copy number variant studies, so looking at these copy number variants, they've used exome studies, which can look at rare single nucleotide variants, and now they're starting to use whole genome studies, and there's going to be a slew of whole genome studies likely to come out and related to autism over the next couple of years. And this has been largely technology driven, and whole genome sequencing will be really exciting because eventually you're going to be able to query every single type of variation in eventually probably 1 million people or something, right? So already we have hundreds of variants that are confirmed, and thousands of variants in hundreds of genes are projected to be involved in autism. So this is a very complex genetic disorder. Now, these genetic variants don't arise willy-nilly. There's a structure to them, and that's related to the genetic architecture of human traits, okay? So what happens basically is that there are two types of variation. There are, not two types of variation, there's a spectrum of variation. There are small effect mutations and large effect mutations. The small effect mutation might slightly change the expression of a gene, for example, whereas a large effect mutation might completely disrupt a gene, right? And what happens is the small effect mutations tend to, so you have, here you are, and here's your entire lineage, everyone who was ever related to you. And what happens is, across time, because of natural selection and other pressures, you tend to keep the variants that had a small effect, and you tend to remove the variants that had large deleterious effects from the general population. So when we look at you, the majority of genetic variation you'll have will be the small effect common variation that you inherited from your ancestors, whereas a minority of the variation you have that might be the most disease relevant, though, will be these large effect variants. And this bears out in what we observe in autism. So for example, Fragile X mutation increases your risk from baseline, the 1% or so, to about 31-fold for autism, right? And what we find is that's either inherited recently, or in a few cases it actually can arise de novo. But basically, Fragile X mutations are very rare, and they are not passed down from your ancestors, like millions of, tens of thousands of years ago. Duplication 15q syndrome is another syndrome that increases the risk for autism about 50-fold. And duplication 15q almost exclusively arises de novo, meaning it's not even in your parents, it just shows up in the person with duplication 15q syndrome. And it can be, in a few rare cases, inherited. And then there's common variants. Now the interesting thing about common variants is I told you they're going to have a small effect, and that's actually what is observed. We actually currently, no single common variant has been identified as contributing to autism. But we know from the types of studies that have been done that the max risk from a common variant is going to be 1.2-fold, which is very small compared to these others. Meaning that if the general risk is 1% in the population, your risk with one of these variants goes up to 1.2%. And of course, they must act in concert with some other types of variation or other common variants to really push you over the threshold for having autism. And more recently, there's been a lot of excitement over deleterious rare de novo variants. So these are brand new variants that are only seen in you and not in your parents. It's likely coming from the germline of your mom or dad. And they've been found in individuals with autism to confer a 2 to up to a 20-fold risk depending on how you look at them. And there's many types of variation that are still poorly defined. So these are deleterious or de novo variants. That means that they are mutations that break a protein. There's also missense de novo variants, which just change how a protein might be structured or is coded. There's recessive variants, which you have to have 2 hits on the same gene. And then there's rare inherited variation. And so there's speculation about all of these. But the difficult thing about many of these other types of variants up here is it's hard to identify single genes that the variant falls on. You have to have very large sample sizes, and those don't exist yet. But already, looking here, we have hundreds of genes implicated. And these types of variants can identify specific genes. So for now, we don't know what's up there, in the old map-making world, if that's what they would write when they didn't know. There's also this exciting area of somatic mutations, where you might have a mutation that just shows up in your cell's developmental lineage, and there, we don't really know what's going on yet either. But these are going to be part of the picture, so we might know hundreds of variants right now. There's going to be way, way more later. So what we're interested in our lab is looking at how that genetic variation ultimately influences the brain. And to understand that, we have to understand what happens at the epigenetic level, at the RNA level, at the protein level, and how these different molecular hierarchies change across development and across cellular subcompartments, major cell types in the brain, microcircuits, and how that eventually affects microcircuits, long-range circuits, and cognition and behavior. And so this is a very complicated task. We don't usually have to deal with this multiple hierarchy situation when we're dealing with cancer, for example, right? There's usually a few cell types that are involved in specific cancers. So what we do in our lab is we're very good at looking at the transcriptome, and we've been expanding to other areas, but the transcriptome is our bread and butter. And the transcriptome is really cool because it can essentially reveal many of these molecular levels of organization to you. Sorry, losing a little resolution there. So if you look at a gene, you can look at genes across development, you can look at them across brain regions, and you can look at them across cell types. And what you'll find is there are certain patterns that emerge for specific genes. So for example, with this gene SMARC2, you see that it's highly developmental. There's fetal and infancy, it's the highest expressed, and then it's decreased in expression throughout. It's not very region-specific, though it might show a proclivity for the cortex. And it seems to be found in astrocytes and neurons over other cells. If you look at another gene, like met-tyrosine kinase, you'll find that it has an opposite developmental trajectory, largely cortex-specific, and seen mostly in neurons. And another gene, OLIG1, has, again, similar trajectory as MET, but it's seen mostly in white matter and in oligodendrocytes. So what you should be aware of from this is that there's 20,000 protein-coding genes out there, and each one of them has a pattern like this. And so we can use these patterns across development, across brain regions, and we can use that to eventually understand what's happening with genetic variants in autism, and I'll show you how we do that. So today I'm going to talk to you about molecular convergence in ASD risk. And I'm going to start with typical cortical development. We're going to go into the autism cortex and then discuss two other approaches using the genetics first approach and network analysis. So we'll start with typical cortical development. And the idea we had, this was in 2013, was to, with all these variants being implicated in autism and specific genes being implicated in autism, could we map autism risk genes to cortical development? And the way we did this is we compiled sets of risk genes. Oh, that is the wrong button. So we compiled sets of risk genes and from multiple sources. So what we did is we took those rare de novo exome variant studies, so where you have autism-related mutations, but ones that are seen only in autism, but not in people who don't have autism. We looked at genes that showed differential expression in autism cortex based on some older work we had done. And then we looked at candidate genes implicated in autism by rare variant studies and other mutational studies. We also looked at genes implicated in intellectual disability. And some of those will overlap with autism genes, but many of them are distinct and only implicated in intellectual disability. So these are all different types of genetic and molecular evidence that are related to autism or related phenotypes. And then we map these to cortical development using gene expression data from the BrainSpan RNA-seq data set. So that's basically expression data that spans multiple brain regions and multiple time points, and I'll show you exactly how we do that in a moment. But the main challenge we have is, as I mentioned, there's thousands of genes in the genome. And if you have all these genes, it's hard to make sense of what they're doing at any given time or place. So what we do is we use the pairwise functional relationships between genes. So what we do is we actually use a statistical association between one gene and every other gene. And what that lets us do is, for example, compute the correlation or an equivalent of the correlation between a gene and every other gene across brain development or across brain regions. And that allows us to connect these genes. So a very strong correlation will result in genes being closer to each other. A weak correlation or a non-correlation will be resulting in the genes kind of being far from each other. And what you see from that is that the genes form clusters. And we call these clusters modules, and we find that they share function. And this has been looked at in many different contexts, not just in the brain. And we can then leverage these modules and this modular structure to this gene network to go from thousands of genes to just dozens of modules. And then we can ask, where are our genes of interest? For example, if we're looking at autism genes, we might ask, where are they? And we might find that they mostly fall in this yellow module. Then, from there on, instead of studying individual genes, we'll be asking what happens with modules. So we can understand module-level biology by basically moving across biological levels. So we could ask, what do those genes do during brain development? So we could summarize the expression level of the yellow module genes in developmental data, in brain region data, and cell types, and see specific patterns that are shared by this module. And we can also ask, are these genes more likely to interact at a physical interaction level by looking at protein-protein interaction data? We can also ask if these genes are likely to be co-regulated by common transcriptional factor. Transcription factors or chromatin modifiers that might act upstream of them. And so these are all bringing in bioinformatics after we initially make this co-expression network from the data at hand. So with the BrainSpan data, we went from 21,000 genes to 17 modules. And these 21,000 genes spanned 8 weeks post-conception to 1 year of age. So we had over 150 samples over these time points. And they were all portable samples. And we were able to go from 21,000 genes to 17 modules. And we confirmed that these modules were reproducible, and 12 of these modules were highly reproducible, found in independent cortical developmental data, meaning that you pull out any cortical developmental data out there, you're going to find this type of structure. And we can leverage this structure now to understand what's happening with autism genes. So what we did is we first started looking at the differential co-expression genes, the candidate genes in autism, and also intellectual disability candidate genes. And we asked, are they overrepresented in any of these modules across development? So each of these modules comprises about 200 to 500 genes. And what we can ask is, are these ASD inherited variants overrepresented in any of these modules? Are the ID variants overrepresented, or are the differentially expressed genes overrepresented in any modules? And we also had differences between these sets. For example, the ASD only set where we take away the ID genes, the ID only set where we take away the ASD genes, and the overlap between DSD and ID genes. And what we found is that there were 3 specific modules where we saw this risk converge. And we call them, very unemotional names like M13, M16, and M17. And what we find here is that every list that has some ASD-ness to it, if you will, the Safari ASD, the ASD only, and the ASD ID overlap, and the ASD M12, we have it M13, M16, M17 enriched, and we did not see enrichment for intellectual disability only lists or the intellectual disability genes as a whole. And so now we can look at what is the developmental trajectory and the molecular function in these modules. So when we look at these modules from post-conception week 8 to 1 year of age, what we can see is that they all increase during brain development. And what's remarkable is they're all higher postnatally, so this is birth, this red net line. They're all higher postnatally than prenatally. And when we look at their molecular function, so this is asking, are known pathways enriched in these modules? And they're highly enriched. In a typical analysis, you expect a z-score of 3 or 4, which is the equivalent of a p of 0.001 or a p of 0.001. Here we're going to very, very small p-values. So these are very enriched for synaptic transmission, release of molecular signals, homophilic cell adhesion, nervous system development, cation channel activity. And what's interesting is these might all be at a superficial level, synaptic development, but these represent different trajectories of different genes involved in different phases of synaptic development. And the interesting thing is when we related it back to what happens in the autism brain at a gene expression level, these were all overrepresented for genes that are found downregulated in autism cortex compared to controls. So basically, these are genes that go up during development, but they're down in autism. Then the other set of genes we looked at were the genes implicated by non-inherited or de novo risk. And so, like I said, you don't find them in the mother, but you find them in the child with autism, right? And what we did is we did this similar. We did it similarly. We scanned through all the modules and we asked, are the protein disrupting ones enriched? Are the missense variants enriched? Or what if we combine them, right? What we found is 2 very distinct modules, M2 and M3, were enriched. M16, the one that I'd shown you before, was also enriched, but these stars actually reflect whether or not it replicates in independent data. So we always do independent replication when we can. And what we found is that we couldn't replicate this one, but we could replicate this very reliably. And looking at M2 and M3, you see that these follow a very distinct trajectory from those initial autism-related modules I'd shown you. So these genes are all downregulated postnatally relative to prenatally. And they're related to molecular functions that are involved in DNA binding, histone modification, more DNA binding here, regulation of gene expression. And it turns out that they contain a lot of chromatin modifiers and transcription factors that harbor these mutations. And the specific genes that harbor the mutations are actually highly interacting at a protein-protein interaction level. And so basically you would expect compared to what you would expect by chance, these genes are 2 to 3 times more likely to interact with each other in a physical sense, which means that they're forming complexes, transcriptional complexes, chromatin remodeling complexes during brain development. And further analysis which I won't go into too much here, basically showed us that these genes were highly susceptible to haploinsufficiency, meaning that if you deleted one copy of the gene, you would get a dosage-dependent reduction in gene expression. And if you looked in human neural progenitors that are being cultured and differentiated, you find these genes co-expressed also. So basically these modules are reproducible in vitro data, alleviating the concern that these could be products of postmortem brain analysis. We're looking at basically frozen brains from frozen postmortem brains. So the other thing is that there are genes that are mutated in unaffected siblings, the controls. So as I told you, there's a lot of background mutation. It's hard to distinguish real stuff versus the background stuff. And so what we know from looking at big sets of genomes is that there's about 100 variants in every individual that are loss of function. And we all harbor 1 to 2 of these rare de novo variants. So even if you're a child without ASD, you can have a variant like that. And in every individual, about 20 genes are completely inactivated. So those are actually often olfactory receptors because they're probably the least important. But there's a lot of genes that have redundant function. So if you're seeing a mutation in a gene, it could just be because that gene's not really important. Or that there's something redundant. So we did the same scanning, but using variants that are found only in controls. And what we found is that they're not enriched anywhere. So basically, we'd identified an autism-specific signature that was related to brain development. And recently, there have been larger and larger sequencing cohorts. So back when we looked at the data, it was 965 families. Now it's 2,700 families. We've looked at it with the newer data set, and we still find the same 2 modules and not the other modules. And related to a rare de novo mutation, that is. And if we look with adding in even more samples, we still see the same 2 modules. So basically, these 2 modules can be said to almost definitively be affected by rare mutations that are found in autistic individuals. But remarkably, we were able to do it with all of that data, right? Now, the other thing is, I told you that these variants are potentially affecting cortical development. So the cortex develops into a 6-layered structure, and we kind of grossly think about it as the upper and lower cortical layers. So layers 2 to 4 are involved in cortical connectivity and inter and intrahemispheric connectivity, whereas layers 5 and 6 go down into the subcortical regions. This is not exclusive, but that's the general rule. And so what we did is we used laser capture microdissection data from the Allen Brain Institute, where they've basically taken the 6 layers of the cortex and dissected out specific cellular populations and done gene expression profiling and identified distinct expression signatures from non-human primates. And the question we could ask now is, having identified our modules, were they more likely to affect certain cortical layers versus others? And there's a general hypothesis in autism that in typically developed individuals, we have a certain inter and intrahemispheric connectivity. In autism, it's thought that there's increased local connectivity and decreased long-range connectivity. And so what we would expect, and what we hypothesize, is that the genes affected in autism are enriched in the upper cortical layers because those are the ones related to cortical connectivity. And so we were able to test that by just asking, how do these 5 modules, which were implicated in autism, these 2 by the de novo variants, these 3 by the inherited variants, how are they enriched across these cortical layers? And so basically, if you go across this line, you're enriched. And what we found is that layers 2 through 4 are indeed enriched for many of these modules, despite the fact that they affect different developmental time points. We could also relate this to the different cell type markers at the gene expression level in the way I kind of showed you before. And what we could see is that the mutations were, or the autism-associated modules were largely related to upper layer glutamatergic neurons. So basically from these analyses, we concluded that ASD genes disrupt glutamatergic projection neurons in superficial layers and thereby affect inter and intracortical connections. There's also some evidence for lower cortical layers and interneurons, but that's not going to be as apparent until we get more gene expression data across brain development and get better, higher resolution modules, basically. So in conclusion from this first part, what we have is that we've identified M2, this developmental M2 and M3, which are early fetal development, and then developmental M13, 16, and 17, which are later fetal development into childhood. And all of these modules show some proclivity for layers 2 through 4, the superficial layers of the cortex. And looking in some additional fetal developmental data that I didn't discuss, that's also dissected across fetal cortical laminae, we find that most of these modules affect the cortical plate and the subplate. So there is an interesting trend in the earliest module affecting the germinal zone. So one other little bit of evidence I didn't discuss is that we also could identify these modules, or that these modules are co-regulated by Fragile X mental retardation protein and also common transcription factors. So based on this evidence, we were able to show molecular convergence in typical cortical development of all these very heterogeneous autism mutations. It doesn't mean that every autism mutation affects those processes, but that the vast majority of them are likely to affect those biological processes. So then the next question we had was what's going on in autism cortex. So we'd done an initial study in 2011 where we looked at 9 autistic individuals and 16 controls. And we'd use a similar network analysis method to go from 8,000 genes to 18 modules. Now the interesting thing here is that this is just looking at 19 autistic individuals. In genetic studies of autism, they've needed thousands of individuals to see the same to basically implicate the same gene. So you need giant sample sizes when you're looking at what's going on at the genetic variation level. But our hypothesis here was that if you're looking at what happens downstream of the genetic variation, what's happening in gene expression, we would be able to see molecular convergence. And we actually did. There were 2 modules that were associated with autism. So as you can see here, this is a module I'd already discussed. And it's down in 2/3 of individuals with autism relative to controls. And this other module is upregulated in autism relative to controls and related to inflammation, microglia, and astrocytes. We were able to take these modules and study additional biology at the module level, and one thing we found is that there was specific enrichment of a genome-wide association signal in M12 right here. So common variants that affect autism risk were enriched in M12, but not in M16. And so this is a way of essentially saying transcriptionally all these genes work together. Together, do common variants affect these same pathways? They do when the pathways are synaptic pathways, but they don't for the pathways that are related to inflammation and microglial function. So back then, as I mentioned, we only had 19 individuals. So we wanted to expand this to a larger set. And so we had some, I think, very obvious questions. First of all, can we replicate what we'd seen before, right? Maybe we got lucky with those 19 individuals. Maybe, it can always happen. And what about splicing and non-coding RNA? So before we'd use gene expression microarrays, and those can only really probe the relative expression levels of genes that are protein-coding genes. And then the other thing we were concerned about were potential confounders, and I'll explain what those are here in a second. So you have effects from age. So people who are younger and have autism might have a different gene expression signature from those who are older and have autism. You have effects from sex. You have effects from medications that the person might be on, and you have effects from seizures, that's the seizure mark. And this involved not just my efforts, but a lot of effort from Grant Belgard, a former postdoc in the lab, and Vivek Swarup, a current postdoc in the lab. And I'll show you which parts they've been involved in. But basically, we first went with the replication question. So we took the fold changes, basically how is every single gene changing between autism and control from our microarray study, and compared them to fold changes from completely independent samples and with a different type of transcriptome technology. And what we found is that there's high concordance at R squared of 0.6 for the genes that were found to be significant in Vonnegut et al. And that's the old study. Compared to the genes that were not significant where the R squared is only 0.13. So basically there's a highly concordant signature that can be reproduced in independent brain samples regardless of what method you use to query gene expression. When we combined all of those samples together, we could look at how these samples cluster. So red here is autism, black is controls. And then here I have the brain region, so we looked at frontal cortex and temporal cortex together. Then I also have age of the individual, the sex of the individual, and the quality of the RNA. And as you can see, it's pretty convincing that, first of all, the autistic individuals are clustering together, they're not perfectly, and these other factors are not driving the cluster. And the general gene expression pattern is there's several hundred genes that are downregulated and several hundred that are upregulated. In ASD, the downregulated genes are related to neuronal function, as we'd seen before, synaptic transmission and calmodulin binding. And the upregulated genes are glial genes, and they're related to immune function and signaling. We also looked in cerebellum, which I don't discuss here, but we didn't see any significant changes in cerebellum, which is a little surprising because there's a lot of focus from certain groups on the cerebellum and autism. And we've really dug into the cerebellum data in a lot of different ways, and we cannot find significant gene expression changes. And there's several reasons for that, which I can discuss afterward, but basically the idea is we don't see. Then what we can do is we can take the shared expression signature of these differentially expressed genes, and we can ask how, first, does it replicate in even more independent samples? So the way we did that is we looked at this current set as the autism discovery set, and these are controls, and we compared them with each other using this gene expression signature, and we find that they're significantly different. Then we used an independent set of autistic samples that were much younger, where we didn't have matching controls. So we held them out from the analysis, and we analyzed them after the fact. So these are like 8 additional individuals. And we find that they share the same signature as the other autistic samples I just showed you, so they're not significantly different from each other, and that signature is different from controls. We could also compare this signature across all the different factors we could possibly measure in these data, right? So we looked at diagnosis, we looked at the sex of individuals, their age, the brain region we looked at, whether or not they had seizures, psychiatric medications, the pH of the tissue, the quality of the sequencing, and also certain diagnostic metrics that are used for autism. And we found that the only thing that there was really association with is diagnosis. That's an extremely significant p-value, and a minimal correlation with age. Interestingly, the ADIR score is known not to be a quantitative metric of autism, so it's not surprising that it's not related to the gene expression signature. We also wanted to evaluate whether or not this gene expression signature was just some decrease in neurons and an increase in glial cell types, which is what you see in Alzheimer's disease. So we compared with Alzheimer's disease. What we basically see is there's a minimal overlap with Alzheimer's disease, and there's no significant overlap in the genes that are differentially expressed in Alzheimer's compared to autism. And that's just kind of a sort of a reality check on, are we really looking at something potentially autism related? Now, I mentioned the non-coding transcriptome. And the non-coding transcriptome is really interesting because most studies over the last 30, 40 years have focused on protein coding genes. The protein coding genes, people will change the number of protein coding genes on you every so often, but it varies from 19,000 to 21,000 depending on what tissue you're looking at and what your definition of protein coding is, believe it or not. So there's many other types of transcripts. So there's ones that don't follow the canonical protein coding structure and read through the end of the transcript. There's pseudogenes which look like protein coding genes but are never translated. There's TUCPs, transcripts of unknown coding potential, which are very amusing, and usually a headache actually. But these also exist in a large number. But there's confirmed long non-coding RNAs. These are RNAs that are known not to code for a gene, or known with all the current evidence at hand. And there's over 60,000 of them actually. I'm only listing the ones from a certain study. And in brain, there's thought to be thousands of them, and in our data we can detect about 3,000 of them. And out of those 3,000, 60 were differentially expressed. And what you can see from here is that the differential expression based purely on non-coding RNAs is sufficient to separate out many of the autism samples versus controls. And what that basically means, and the reason that's important, is that many people don't know if these non-coding RNAs have any function. And so it's unlikely that something that doesn't have some important function is likely to be differential between disease. And there's additional evidence we can pull into, because the reason these guys are hard to study is because we don't know much about them. We don't know much about the protein. There's no protein they code for, right? So what do they do? So one way we've looked at some of these long non-coding RNAs is looked at them from an evolutionary perspective, and we find that the majority of them are primate specific. So if you look at, this is an autism sample and this is a control sample. This is cerebellum, temporal cortex, and frontal cortex. And what you're looking here at is a non-coding RNA upstream of a coding gene. And you can see in controls there's not much expression where the exons of this non-coding RNA are, right? But if you look in autism, you see that there's higher expression in the frontal and temporal cortex relative to this control sample. And there's, iffy expression in the cerebellum. So looking down here, what you see is that this non-coding RNA is conserved in chimpanzee and rhesus macaque with a little gap here, but not really in mouse at all. We can even look at expression data from these species and see that this non-coding RNA essentially does not exist beyond primates. Additionally, looking at this, the specific non-coding RNA, looking at it in autism, you see that it's overexpressed in autism. And if you look at it across development, birth being about here, you see that it's a prenatally expressed link RNA that's downregulated postnatally. So here you have a long non-coding RNA that was supposed to be downregulated during development, but it's seen as upregulated in autism. The other thing we've looked at quite extensively is cortical patterning in autism. So what I mean by cortical patterning is that certain genes are going to have a preferential expression in frontal cortex compared to temporal cortex and vice versa. And we see 551 genes that are either higher expressed in frontal cortex, this set of genes, or higher expressed in temporal cortex. And when we look at autism, we only see 51 such genes. And we'd seen this in our previous study too, but we were able to expand on it here. And what you can see looking just at this heat map, right, is this was a red solid block and a relatively blue solid block here. It's splotchy. Same here. So basically you're losing the signature that distinguishes frontal cortex and temporal cortex. And we ask, for the genes that are cortically patterned in controls but not in autism, what are the pathways that are implicated? And these pathways include regulation of certain signaling pathways, but with receptor signaling, which is known to be involved in cortical patterning, negative regulation of cellular differentiation, and tissue development. And so we have the question, maybe there's an upstream transcription factor or something that is essentially promoting cortical patterning in controls, but is dysregulated in autism. So we did a scan upstream, 1,000 base pairs upstream of all these 523 genes compared to the entire genome. And we found that there's a motif for SOX5. We also found a motif for 2 other transcription factors. But we went with SOX5 because it turns out SOX5 also exhibits attenuated patterning. So these are pairs, a line here connects the same individual for temporal cortex and frontal cortex. And what you can see is that in controls, the frontal cortex is consistently higher in expression of SOX5, right? But in autism, a good fraction of the individuals are either flat or going in the opposite direction. When we looked at the correlation between SOX5 and its targets, we found that in controls there's a negative correlation, about minus 0.24, whereas in autism there's no correlation. And what that means is that we would predict that SOX5 represses these targets. And this is actually confirmed by a lot of the SOX5 literature. It plays a role in repressing certain developmental genes. And if we look at every gene that's not a target, we don't see any difference, right? So basically this is a target-specific patterning loss that is related to SOX5. And other evidence out there has already implicated SOX5 in postmitotically regulating migration, postmigratory differentiation and projection of subclade and deep layer neocortical neurons, sequential generation of corticofugal neuron subtypes, and the Wnt signaling pathway is known to be regulated by SOX5 and is implicated by mutations in autism. So a lot of this kind of fits with the picture of what we've seen before, but identifies a specific factor that might be driving some of these changes. The other thing we wanted to look at that we haven't looked at before at all was alternative splicing across these samples. So we looked at, actually we had looked at alternative splicing previously, but only in 3 individuals with autism. So here, the way we quantify alternative splicing is we look at RNA sequencing reads, and we can identify, if we have 3 exons here, sometimes this exon will be left out of the eventual isoform that's coded. The way we can quantify this is we can look at RNA sequencing reads that span the junction between this included exon and ones that skip this junction. And we can identify many splicing events like this, including skipped exons that are included or excluded from the 5' end of the transcript, the 3' end of the transcript, or when one exon is chosen over its neighbor. Using these measurements of these events from our RNA sequencing data, we again did the same differential analysis that I showed you before, and we find that there's a splicing signature that groups autistic individuals away from controls. The interesting thing about the splicing signature is it's absolutely neuron-specific. There's no signature of alternative splicing that's different in autism in microglial genes or astrocyte genes or oligodendrocyte genes. It's entirely in neurons. And looking at the pathways, There are secretory pathways, neuron projection-related pathways, pathways related to biological adhesion. Basically, the proteins involved in these pathways are known to require specific isoforms to carry out specific cell-cell recognition and different biological processes. The other interesting thing is that this signature is distinct from gene expression, meaning even if we took away any even mildly differentially expressed gene at the gene expression level, we're left with hundreds of genes that have alternative splicing that wouldn't be detected otherwise. And so this is a distinct signature from whatever we identified previously. And these changes are, again, not seen in cerebellum. Another interesting thing that arose from these splicing analyses is the role of microexons. And so we worked with collaborators in Canada, Manuel Arimia and Ben Blanco, to look at what were the sizes of the exons that were dysregulated in autism. And what we found was there was a preponderance of small exons. So most alternative splicing studies actually look at exons that are more than 27 nucleotides, actually more than 50 nucleotides most of the time. But there's these tiny nucleotides, which are 3 to 15 nucleotides, 16 to 27 nucleotides, which are disproportionately misregulated in autism. And there's a known regulator of these microexons, as they're called, that is also dysregulated in autism. And when we looked at the events that are regulated by that known regulator, which is called NSR100 here, but it's also known as SRM4, we found that most of these events could be explained by ones that were regulated by NSR100, and that NSR100 targets were correlated with the expression level of NSR100 genes in our brain samples compared to ones that were not regulated by NSR100. So essentially we have dysregulation of this gene, SRM4, and its microexons that it regulates. And we also have support for other factors that could be driving the splicing change, which I won't go into here. We've done developmental studies on RBFOX1, and we also found in Vonnegut et al. That RBFOX1 is misregulated. We also have additional evidence for another splicing factor, NOVO1. And what we believe might be going on here is that multiple splicing factors, although there might be one splicing factor change in one individual, another one in another individual, they might be changing splicing in the same genes. And again, this is a different route to convergent changes in autism. The other thing is a genetic first approach. So what I've shown you so far is all in idiopathic autism. That means we don't know the cause of autism in the individual. But we did have a subset of individuals, 8 individuals, where we knew the cause. It was duplication 15q syndrome, that one that I told you increases risk for autism 50-fold essentially. So the question that we had is, if you have the same genetic hit, duplication 15q, do you have a more homogeneous gene expression signature? And will your transcriptome in this genetic subtype look like the general idiopathic autism transcriptome? So looking at duplication 15q syndrome, the way it's specifically defined is there's an extra piece of chromosome 15, the 15q region, and it arises de novo most of the time, like I said, because it has essentially a 90% penetrance for autism, and it's associated with developmental delay, learning and cognitive disability, seizures, and dysmorphias. So we first looked at transcription in the 15q region, basically where this lesion is in these individuals. And so looking at this region, what we did is we looked at duplication 15q individuals relative to controls and autism relative to controls. What you see here in blue is duplication 15q, falling between the range. Many of the genes fall between the range delineated here, and one here is a 2-fold change because it's a log 2 scale. So basically many of these genes are doubling in expression or close to doubling in expression in duplication 15q, but not in idiopathic autism. Idiopathic autism, wherever the changes are significant, tends to be in the opposite direction. So that's interesting because it's, first of all, it tells you that whatever's going on as a primary thing in idiopathic autism is definitely not the thing that's going on in duplication 15q syndrome. And this work was largely done initially by Grant Bellegarde. Then looking at the duplication 15q transcriptome outside of the 15q region, what we find is indeed all these duplication 15q region individuals in blue here cluster together. They cluster completely away from all the controls. And they have similar gene ontology, the molecular functional term enrichment, and cell type enrichment as idiopathic autism. And when we look at them, we can compare idiopathic autism versus control fold changes to duplication 15q versus control fold changes, and we see that they're highly concordant. And there's a slope of 2.0, meaning that when you have a 2-fold change in idiopathic autism, it tends to be a 4-fold change in duplication 15q syndrome. So we do see a more homogeneous or stronger change at the transcriptional level in this genetic subtype of autism. And interestingly, this also recapitulates at a splicing level. So whenever we see, for example, a 40% change in the inclusion of an exon, it tends to be a 20% change. It tends to be a 50% change in duplication 15q syndrome. So basically, what these analyses tell you is that in a type of autism where we know that there aren't environmental factors at play, and we know what the lesion is, and we know that it's independent of all the confounders and stuff I showed you before, we have this highly concordant gene expression signature with what we see in the rest of autism. Doesn't mean that every single idiopathic autism individual has a genetic lesion or a combination of genetic lesions only, but that's a defining molecular signature of the majority of autism. So the other thing we've done is used our network analysis methods, and network analysis has allowed us to go from the 2 initial modules we identified in Vonnegut et al. To many more modules now. Well, many more that are autism associated. So basically, ASDM12, the one that was downregulated in our initial study, splits into 4 modules that are downregulated not just in autism, but also in duplication 15q syndrome. And the M16 splits up into 3 modules that are upregulated not only in idiopathic autism, but also in duplication 15q syndrome. So now we have genetic evidence from Dup15q supporting every one of the modules that we looked at before in idiopathic autism. We can look at specific modules and gain some interesting insights. So if we look at M1 versus M5, so these are different M's. As I told you, there's one conundrum we often have is, like, as we do different studies, what do we call the old modules? So I didn't fix that issue here yet, but the idea here is these are different M1s and M2s from what I referred to before, and I'll call the old ones developmental M1 and M2, or ASD, you'll see here in a moment. So if we look at M1, it's similar to ASD-M12, and it contains genes that are down in autism and duplication 15q syndrome. They're enriched for autism and schizophrenia and genome-wide association signals. And we have genes that are neuronal genes, synaptic transmission, calmodulin binding. M5 is similar to ASD-M16, and it contains genes that are upregulated in ASD and Dup15q. There's no genome-wide association signal, just as we'd seen before. And we see enrichment for microglial and inflammatory markers. And the interesting thing about them is these 2 modules are highly anti-correlated with each other across individuals, meaning that if an individual has high expression of the microglial module, they tend to have lower expression of the synaptic module, suggesting that these modules might be related to a neuron-glia interaction, a neuron-microglia interaction. And we've been wanting to investigate that and have started doing so. The other interesting module is M2, which is an early chromatin regulation and long non-coding RNA containing module. So M2 is not changed in adult autism cortex, but it was still interesting. The reason is that it has a 2-fold enrichment for the rare de novo variants I told you about before. And a 5-fold enrichment for developmental M2, so that was this module that I'd shown you earlier in the talk. So basically these genes, developmentally, are still seen as co-expressed in adult autism, but they're not different necessarily between autism controls and cortex. And so this module could represent something that happened very early in development. And what I thought was the most interesting is this module has a 5-fold enrichment for long non-coding RNAs. And when you look at its gene ontology, it not only includes these pathways related to chromatin binding, but also things related to the regulation of gene expression and mRNA processing, which are functions that long non-coding RNAs are known to be involved in. So I think one of the really exciting things we'll be able to do in the near future is go back to developmental data, make a better developmental data set, and figure out what are these long non-coding RNAs doing in brain development, and how are mutations potentially affecting them and contributing to autism. Because it's thought that these long non-coding RNAs, although we don't know their function, might be just as important as the most important transcriptional regulator that differentiates neurons. The other thing we can do is we can compare the expression signature at a module level between idiopathic autism and duplication 15q, and you see that most things follow this blue line, and there's again this high concordance and this slope where we tend to see a stronger change in Dup15q. But an interesting module is M13 here, which is out of the sort of expected line. And so what we looked at M13 and we found that it was not enriched strongly for cell-type-specific genes. There were no genes in it that were related to that were in the 15q region. However, we know that translation-related genes UBE3A and CYFIF1 are upregulated in Dup15q syndrome, and we find that the genes in this module are highly related to translation. So for all these RP genes are ribosomal, are RNAs that code for ribosomal proteins. So what we think is that, and we're trying to find additional evidence on UBE3A regulated targets and cyclic-1 regulated targets, but basically we think that these genes might be the first things to go awry in Dup15q syndrome, and then downstream you get the other modules that we were showing you that are important with idiopathic autism. So what I've shown you here can be, I think, synthesized in an interesting way. So I've shown you this graph of how there's a relationship between how long a mutation has been in the human population and how pathogenic it's likely to be, right? And what I've also shown you here is that what we found in these modules kind of reflected that, right? So we had these developmental modules, M2 and M3, and this sequencing module, M2, that were enriched for the rare de novo mutations. And then we had these ASD M16, the developmental synaptic modules, and the new synaptic module that are enriched for common variants or inherited variants. And so what it looks like is that the more severe mutations are affecting early brain development and then eventually filtered out of the population because of that. And these milder mutations are affecting their potential downstream targets. And this fits with what's kind of known from the Big ENCODE project, which really didn't study many brain cell types. But what they found is that many molecular, using what ChIP-seq data and a lot of other regulatory data, they could divide up the genome into kind of 3 molecular levels of regulation, and they found that there were different properties in the top tier regulators and the bottom tier regulatory targets and the intermediate tier of molecular players. And so the idea here is that these guys are big time, like transcription factors, chromatin modifiers. They affect these genes, and then these genes change these genes downstream. What we seem to be seeing is that the developmental M2, M3, and the sequencing M2 are related to this top tier of regulators, and these guys are these bottom tier of regulated things. But there's a whole spectrum of how this works, and it'll be interesting to connect this genetic architecture to the molecular regulatory architecture. Now, this molecular regulatory architecture, unfortunately, largely comes from cancer cell lines. So what will be important is to understand how all these, there's many top-tier regulators that are at play in multiple stages of brain development. There's different transcription factors that determine the next step in differentiation of cells and then specification of cellular subtypes. So what's going to be really interesting is in the near future, we can make a map of regulatory networks during neural cell lineage specification. There's already projects that are aiming to do that. And what we can do from this is begin to understand how the basic approach I took here to look at, gene expression and how it's affected by these mutations at a whole tissue level is actually affecting individual cell types as they differentiate in brain development. So I hope what I've conveyed to you today is that ASD genetic risk converges during cortical development. I hope that's pretty definitive to you. That this is specifically related to early transcriptional and chromatin regulators disrupted by ASD-specific mutations in normal cortical development. And then I've also shown you that there are shared changes in ASD cortex supported by the genetics of autism. And this includes newly characterized dysregulation of long non-coding RNA and splicing, duplication 15q syndrome recapitulating the idiopathic autism transcriptome, and co-expression networks that identify new biological pathways that are implicated in autism. And I'd like to acknowledge the guidance I've received over the last 5 years from Dan. This is a very complicated area of biology. I basically knew nothing about it coming in, so I learned everything I know from him. And then we have lots, we have a large lab, as many of you probably know. I've had help on various projects from different people. So this group of people really helped out in the cortical development project. Jason and Luis have been involved in everything. We work in parallel on different things. And these guys have been involved more in the more recent projects. And we have collaborators in network analysis, Steve Horbach, keeper of time, and Dan, finder of networks. That's if you haven't seen the NOVA special with Dan looking around for networks in Ronald Reagan, it's worth seeing. Then we have the BrainSpan consortium that was involved in generating the developmental data that I showed you, and the microexome analysis, which was aided by our collaborators in Canada. And we've had funding, generous funding from the Simons Foundation, from the MSTP, and multiple NIH grants. Thanks.
questions. I'm sure there are many, and before we take the first question, there will be a reception afterwards in Uganda, so please stop by there. Please.
So these modules that you see that are misregulated, do you think that there's any way to boost up or boost down transcription of all these genes together at sort of a critical time? Is that science fiction? Or?
Yeah, no, I don't think it's science fiction, but it's potentially more complicated than it might seem from this, in the sense that these are modules that are constructed from whole tissue profiles, right? So it might be important to get down at that more cell type specific level before we can really, I mean, we can make cell type specific inferences, but that doesn't mean that that's, if we had cell type specific data, we would get down to a resolution that is not seen here. We only have a certain level of resolution. So the conclusions I made are, like I said, not applicable to every single individual with autism, but they are applicable to many individuals with autism. So I would be hesitant to kind of start thinking of like autism-wide approaches until we have that higher resolution data. Now, the interesting thing about changing the expression of many of these genes simultaneously and at a critical time point is that it might be more worthwhile to pursue some of these modules that are regulated downstream later on during development rather than the early ones, because it seems that some of these mutations really affect prenatal development, but that there are modules that are affected downstream that are potentially more amenable to modulation in adults, for example, right? And we know from things like Fragile X syndrome that it's possible to alter behavior and neurodevelopmental disorders in adults, so I think it is possible. But it's far away. Yes.
Neil, was there a relationship between the physical location of the long non-coding RNAs that were differentially regulated and your genes that were, and transcripts that were protein coded?
Yeah, that's an excellent question. So the general answer is yes and no. So there is, for a couple of them, they're like upstream or downstream of misregulated genes, but it's not above chance, you know? So it's not like the long non-coding RNAs are always upstream or next to a gene that's also differential in autism. And part of the reason for that is you might be thinking about how a long non-coding RNA might regulate gene expression, right? And that being in physical proximity and changing the local chromatin structure or binding to a local transcriptional complex is only one of many mechanisms that are implicated for long non-coding RNA. So it's not surprising that we don't see that as a major pattern.
Was the SOX5 upstream, was that only for long non-coding RNA?
Sorry, that wasn't clear, I think. That was for both coding and non-coding RNAs.
Oh.
Yeah. So that cortical patterning analysis included both coding and non-coding RNAs. Yes?
So your upregulation of the microglia transcripts, does that suggest neuroinflammation or downregulation?
So I think we know from other studies that there is an activation and infiltration of microglia in certain brain regions in autism. They're very small studies. So I think this is reflecting that to some extent, that it's essentially increased microglial signaling and potentially increased microglia. The interesting thing about microglia is you need relatively few of them to do a lot in the brain, right? So when you consider the total pool of RNA we have, they're actually probably contributing very little RNA to that pool. But the fact that we see this upregulation suggests that maybe the signaling in these microglia is really jacked up, basically.
So you think neuroinflammation closely?
Well, what the genetic evidence suggests is that the enrichment for the common variation, for example, and even for the rare de novo variation was largely in the synaptic pathways. So at some level, what this is suggesting is that the neuroinflammation is reactive because we don't find any genes or any gene sets enriched in the neuroinflammatory pathways. That being said, in some of the other neuropsychiatric disorders where they have much larger data sets and much larger sets of common variation, the neuroinflammatory pathways have emerged as a potential player. So it might be the case in the long term in autism. And there's just some complications in studying certain regions of the genome that are related to inflammation that really preclude looking at some of the most interesting genes in genetic studies, so we might be missing it because of that.
You indicated you were surprised by the lack of significance in the cerebellum.
Yeah.
How do you interpret that? Does that tell us something about, maybe that the tonality of the cerebellum is very different, it's not complicated enough, or how do you interpret that?
Yeah, so one thing we know is that the majority of cells in the cerebellum are granule cells, and when you consider the pool of RNA, it's potentially responsible for granule cells are potentially responsible for 90% of the RNA we're looking at. So this really means that in the cerebellum we might have to do more cell type specific analyses before we can really rule out that the cerebellum is not changed at a gene expression level. And that would be kind of the first place, I would think, and so that's why I didn't really dig into it much more yet. Yes.
Okay, so fantastic, obviously, this is really interesting. So I'm, of course, really interested in your overlap analysis of the 15q and the idiopathic autism. And I was curious how you treated the 15q CV and that overlap analysis. Did you exclude the genes from that region?
Yeah, so in that overlap analysis, we can exclude them or not. There's essentially, and you get the same result. So there's essentially like 2,000 genes dysregulated in duplication 15q syndrome, more than that actually. And the ones in the 15q region are only maybe like 30 of them. So you can take them out and you see that exact same clustering I showed you.
Okay.
Yeah. Any more questions?
Well, then, please turn this over and come back.